a demo later, so if you want to check it out, let's go poster. And with that, I'll open it up for questions.
You've got to be motivated to ask questions. Fantastic. Why wouldn't the pharmaceutical industries push a lot of information about their drug-gene interactions? Because it's in their interest to have their compounds used in the best way.
So maybe they wouldn't push it publicly for other people to use, but these pharmaceutical companies also produce useful pharmaceuticals, and if they can, so what I try to do is make it so right now all these networks are pre-generated, but what I want to do is make interface so that you can just generate these networks like on the fly. So these pharmaceutical companies can also use this to enable their pharmaceutical products.
Okay, and then the second question that really relates to the computational work you're doing. What data do you accept straight on without having to worry about is it valid or not? Is there such a thing as top-tier data that would convince you we have to use this data for building our interaction networks?
So all the evidence comes from the stitch and string knowledge bases, and so the way it works is it's not just like, either we include this evidence or we don't. The way it works is actually if you look at these scores over here, you can see how little content they have. So the higher the score, the more content they have. So you can actually filter out, if you don't want low quality evidence, you can filter out what you want. And these data channels also help with that because perhaps you don't consider some of these kinds of methods useful, but you can actually filter them out.
Good, great talk. Thanks.
Thank you so much.
So you deal with the known a lot. You deal with things that people know. You deal with things that are thought to be accurate. When you're dealing with your drug screening, you're dealing with cancer cells in a dish, and you're simply putting drug right on top of them. So there's an enormous amount of evidence that when you change 3D culture types, in vivo studies, when you add more cell types involved like you find in the body, that the responses to drugs can be incredibly varied and often quite different than those we see in culture dishes. How are you going to take that into account when you're thinking about building networks based on drugs in a dish?
So I think this is more of a systematic problem with how we look at drugs and try to figure out drug-gene relationships, because when working in a dish is far different than working in a human. So the only thing I can do is give you enough information about what's known right now, and let the researcher and the biologist who knows more about how these things work, figure out which is compelling and which is not.
So quick follow-up then. You talked about TMZ. The researchers at TGen. First question, did you come down and talk to them? Did you talk to Dr. Behrens and Dr. Drew about their experiences with TMZ, the model systems they've used, the opportunities that they might have to look at the data that you produce? Did you have those conversations? Did you begin them this summer?
I have not yet, but I plan to. So I generate all these networks, but I'm still in the validation phase of, I want to show these networks to researchers and ask them, does this look right to you? Do you think that these intermediate genes work? Or these intermediate genes you accept? And so on. But yes, that is for sure the direction.
Hi, this is a great talk. I'm curious, when you create a tool like this, in what way do you, when it's ready for prime time, how do you make this available to others, and how do you make it discoverable for people to use?
Okay, so one way is, we publish this ACTRP data, and that's one way to use the advanced networks. And another way is, I mentioned a little bit before, I want to make it so you can compute these evidence networks on the fly. And so what I want to eventually do is advertise it so that people can just go to the web interface and they have some drug gene pair that they're interested in, and they can just type it in and then get that evidence network like that. Advertisement-wise, we're talking about going to the Tran Bio conference in Indiana, so I think there'll be a lot of networking to advertising. That's our